Assalamu alaikum wa rahmatullahi wa barakatuh. Good evening, dear professors, dear colleagues, dear students, and dear chairpersons. Thank you, Dr. Ahmed and organizing committee for giving me the opportunity to sharing in this amazing event. Our talk today is under the title of Artificial Intelligence-Based Network Pharmacology and Bioinformatics in Drug Development, Applications and Challenges. Clarivate Analytics and Web of Science Core Collection, one of the most trustable citation index database around the world, and it contains different databases. When I search about the keywords related to this talk, network pharmacology, bioinformatics, artificial intelligence-based network pharmacology, network and system biology and omics, I found more than 64,000 results which distributed especially in the last 5 years with increasing trends. And there is 740 highly cited papers and 23 indexed in the hot papers, with 20,000 publications under the categorization of pharmacology and pharmacy. So it's worthy to know what is network pharmacology. The content today will include a conceptual introduction to network pharmacology and bioinformatics in relation to drug discovery and development, artificial intelligence applications in network pharmacology and bioinformatics, bioinformatic databases, including types and sources, translational methodological designs and workflow used in network pharmacology approaches and bioinformatic tools, challenges facing AI-based network pharmacology and bioinformatics applications, and future perspectives and opportunities for AI-based applications. For several decades in drug discovery and development journey, the authors and the scientists, the researchers, the pharmacologists go in one theory and one approach. One drug, one target, one therapeutic approach, key and look appearance, or the magic bullet to treat all the entities of a disease as immunotherapy to treat different types of cancers. However, they found about 90% fails in clinical drug development with limited efficacy and therapeutic effects because there is complex multifactorial diseases with diverse biological processes and molecular targets. At the end of the 20th century and ongoing, the information technology revolution has been developed with the post-genomic era and advancement in the computational biological techniques, omics technologies, and in silico store, and emergence of the network biology, which is a complex network that related different biomolecules in our body with each other, and to show the interaction between them, about the genes, about the proteins, about the metabolism. So the scientists think about why we use just one drug. We can use polypharmacy or more than one drug to cover multiple targets and multiple therapeutic approaches. So, innovative approach or strategy in drug discovery called network pharmacology was released. Hopkins in 1927 invented this title, network pharmacology. Network pharmacology is an approach or strategy in drug development. So it is combined the drug information in pharmacoinformatics with the biological informatics, bioinformatics, and the biological system together using computational tools or using computational biological tools. This approach helping in defining the drug target interactions and pathways. So we are also need to know a broad categorization of drugs filled in the ethnopharmacology, which is plants, bacteria, and the organisms used by different cultures and different ethnics. We defined network pharmacology. So what is bioinformatics? Bioinformatics are developed in 1978. It is, in simple word, computational biology. There is computational tools to solve biological problems, including several untreatable diseases and several limitations in the drug efficacy and adverse effects. It's a field of science that uses computer, databases, math, statistics to collect, store, organize, and then analyze Analyze these data to what? To show interactions between these data. And so there is also what we call nanoinformatics, which is related to the nanoscale of the drug molecules. What are the analytical bioinformatics tools in network pharmacology? Simply, it's AI tools and applications. Artificial intelligence application drug discovery and development, including predictive modeling in drug efficacy, toxicity, adverse effects, pharmacokinetics, in different models of the diseases. Also, it including AI algorithm analysis to large data sets to identify drug targets and drug-drug interactions in virtual screening, also by using it in rational drug design and repurposing. I think the definition of artificial intelligence was covered in the previous lecture, and I think also if I said that artificial intelligence, most of them used in the pharmacology here and network pharmacology based artificial intelligence is machine learning and deep learning. This is an example of using artificial intelligence in analysis. Here there are different levels of the biological system, including macro levels and the micro levels. And there is machine learning and then deep learning and then leading to network positioning and network navigation and disease and syndrome related predictions. Bioinformatic databases have several types, including genome, sequence structures, microarrays, chemicals, pathways, and enzyme disease literatures. However, the sources is divided into primary and secondary. Primary, it contains original data from the publications or entered in these databases by the researchers. Secondary databases are connected to primary databases and customized by the researchers, even manually or automated. Bioinformatic databases related to network pharmacology, including drug databases or herbs databases, including PubChem, drug banks, and traditional Chinese medicine bank, so gene and proteins, string and uniport, and to the diseases, gene code, and TDDs, and functional annotations. Functional annotations meaning we relate the function of the genes and the protein sequences to their sequences. Integration of the multi-omics data here is an important database to investigate it through application of the AI technology and network pharmacology. What is the definition of omics? Omics is the constituents, all constituents inside the cell. But when we talk as a science, we talk about identification, description, and knowing the function of every biomolecules inside the cell. And this is, can be categorized into genomics, transcriptomics, proteomics, metabolomics, and cytomics. Genomics to the genes, transcriptomics for the RNAs, proteomics for the proteins, metabolomics for the metabolism, and cytomics for the cells. All of these databases give raw data sequences to the bioinformatics to be furtherly analyzed and used in the network pharmacology. These databases are found in each database related to the omics. As an academic researcher and a physician in drug discovery and development journey, where AI-based network pharmacology and bioinformatics could be applied to all stages of the drug discovery, from the discovery and development, preclinical, clinical phases, and FDA review and FDA post-marketing surveillance. So the benefit rules of AI in based network pharmacology, identifying novel drug targets and time, understanding and signaling the pathways of disease types, designing experiments based on drugs and targets, therapeutics for multi-gene dependent diseases, studying drug resistance, maybe antibiotic resistance. Also in clinical trials and post-marketing surveillance. In clinical trials, they act on regulatory frameworks, integration and validation, and adverse event monitoring and management, including the electronic databases that collect these data. AI's impact in personalized medicine, they enhance the patient care through identifying specific patients, characteristics, predict disease outcomes and drug responses, personalized drug selection, and dose optimization. Translational methodological designs using network pharmacology approach in drug development research. This is an important question because till now, it's a challenging knowledge gap. There is no standardized workflow or methodology to use in these types of research. So I collect one of the most published protocols and workflow in the literature, starting by drugs as Chinese herbs. In Chinese or we don't know the exact product or the exact component, so we can search on these components, okay? But if we start with a known drug, so I know the active component. By knowing the active component, I will use other databases to know these components can act with each target inside the cell. So, and also think about my disease that I will go through in my research. This is I will search by different databases on disease targets. Then I will collect the two targets together and making an overlapping and sharing the common targets. These common targets I will search on protein-protein interactions between them and between component target disease networks. This will lead to the bioenrichment analysis, which search on overexpressed genes and overseeing proteins that is related to the phenotypical changes in the disease, okay? And I will go to do another way of analysis by validation. Validation by molecular docking, in which we give the proteins as receptors and our targets of the molecules and give it together. And if there is interaction, so this molecule is acting on this receptor or protein. Also, it will validate it well with in vitro experiments and in vivo experiments. Some publications in the clinical trials using reverse frameworks. They don't start from the drug discovery. They start from the clinical trials. They collect the blood samples from their patients and become to analyze these blood samples on genes and on proteins in proteomics or genomics to reach the dark genes that they can examine, and after that by different drugs, okay? Here, the gene GEO databases, next generation sequences, reaching to the hub genes, which are genes that is mainly correlated with the most genes inside the database, and then validation. These researchers published from our lab, pharmacology, clinical pharmacology in South Carolina University, and we start to use bioinformatics and network pharmacology. All of them are published in Q1 pharmacology journals, according to the Web of Science. Challenges in AI-based network pharmacology and bioinformatics is related to the data quality and the integration of these resources that may be introduced by the researcher itself. Okay? Also, the ethical consideration and legal implications about how can we trust data released from artificial intelligence, okay, without validation. Also, computational illiteracy about the researchers. اسمحوا لي هنا بقى اتكلم باللغه العربيه لان السؤال ده انا بوجهه لاساتذتنا الافاضل اللي موجودين دلوقتي معانا. Are drug researchers using AI-based network pharmacology and bioinformatics acceptable promotional scientific work in national medical organizations and institutes? sorry. يعني ده انا اعتقد حضراتكم محتاجين تبصوا عليه لان يعني دي new era. What are the future prospects and opportunities? Personalized medicine advancement, improvement in drug designs using next generation technologies and interdisciplinary collaborative research opportunities. Conclusion, AI-based network pharmacology and bioinformatics play crucial roles in pharmacology, enhancing various aspects of drug discovery, development, research, and the clinical practice toward improving patient outcome and advancing healthcare. In drug discovery, AI assists in identification of potential drug compounds and suitable patient populations, leading to more efficient and targeted therapies. AI enables personalized medicine and increases efficiency in pharmaceuticals industry. Several challenges are addressed in applications of AI-based network pharmacology and bioinformatics, which need to be explored, analyzed, and modulated by suitable actions planned by different research and industrial and ethical authorities and organizations. Thank you.